This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Really wonderful to be here. I think it's such a treat to do mini medical school. The audiences are always absolutely wonderful, so I'm really thrilled to have the opportunity to do this. My name is Joel Kramer. I'm a neuropsychologist in the Memory and Aging Center in the Department of Neurology here at UCSF. We're also have Saul Villeda here. Saul got his postdoc at Stanford in 2010 and has been pioneering some of what I think is some of the more creative and important work that he'll describe to you next. And then I'm also delighted to introduce Christine Yaffe. I've had the honor and privilege of working with Christine for over 25 years now. In spite of how young we are, we have a long history of working together. She's the Vice Chair of Research over in the Department of Psychiatry here at UCSF. She's she's the head of the geropsychiatry program at the VA Medical Center and runs the memory clinic there. And sort of as a testament to her prowess in the field and leadership, she was targeted or tagged by the Institute of Medicine to be the co-chair of their recent panel on cognitive aging. So we're here to say a little bit about normal and abnormal aging in the brain. And I understand that our colleague Michael Geschwin was here last week laying some of the groundwork for this whole aging and the brain stuff. But we're going to reiterate some. All right, so my basic disclosures: funding from NIH, Hilmblum Foundation, Tau Consortium, and Pearson Incorporated. My bigger acknowledgement here, of course, is the entire Memory and Aging Center. I'm guessing that Michael Geschwin showed this slide last week. This is the entire team over at the Memory and Aging Center. This was from our retreat from a couple of weeks ago. And then the real acknowledgements has to do with sort of a handful of research coordinators who really do all the aging research that happens at the UC. And we're missing a couple of the folks who weren't available for this photo today. Some of you who may be involved in the normal aging research programs at the Memory and Aging Center might be familiar with one of these absolutely fabulous research coordinators. All right, so let's take 1,000 or so people ranging in age from 20 to 90 and give them a memory test. So the memory test is a word list, and you give the folks several opportunities to learn the list. And then 20 minutes later, you come back and say, all right, what were the words on the list? And you look at how well people performed as a function of their age, and this is pretty much what you get. This gradual decline in memory performance the older you get. So this slope here has been replicated in a wide variety of cognitive paradigms. So this is memory, but if you did information processing speed or multitasking or sort of visual spatial problem solving, you'd get pretty much something similar. How many people actually believe that this is true? How many people actually feels that this jives with their own experience in life? Right. So I call this the myth of cognitive decline or age-associated cognitive decline. And, granted that, we all experience some changes with age, but this regression line, this slope, this is an average that doesn't actually capture all the real data. So let me show you what the actual data points on which this particular graph was based. Again, these are the real data, all right? So basically, you've got this 90-year-old who's remembering a lot more items than a 20-year-old, right? So what is this really telling us about aging? Well, first of all, we have to acknowledge that this is a cross-sectional study. It could be that this person was always awesome. It could be that this person was always terrible. And what we really want to know is, well, if you take the same subject and follow that subject over time, what happens to their memory? All right, so we've done a little bit of that as well. So in our program, we have longitudinal memory data on hundreds of people over multiple time points. And if you actually do the plots, this is what it looks like. Right? All right, it looks a little bit like spaghetti or pickup sticks that our kids and grandkids play. But let's start using some statistical modeling to tease apart some trends within this overall gobbledygook. And what you can see here, and this is one approach where we divided this group into 4 subgroups based on the pattern of their memory performance over time. And there are a couple of things that I think are really quite striking, one of which is that there are 2 groups who actually seem to get better over time. Now, I'm not going to argue that their memory is truly getting better. What's happening is that they get this memory test every couple of years, and after a while, after you've done this a bunch of times, you get a little bit better at it. It's just basic practice effects, and this is what we're probably seeing here. But if your memory is good enough so that you can benefit from practice effects, that's a pretty good thing. Then you've got one group here that really starts off kind of low and doesn't change a whole lot. And in fact, this is probably 7 or 8 years later, and you can see this group is performing just about the same. And there's really only one of these subgroups that seems to be declining over time. So these 3 groups all start at about the same level, kind of average for people in their 60s and 70s, but this is one group that's getting a little bit worse over time. And the question really is, what's different about that group? Why is that group getting worse? And what is it about these groups that make them so stable? Because in fact, it really is possible. We have so many subjects in so many different studies that are followed longitudinally that not only show stable cognitive performance, but when you look at their brain scans, you really don't see much change over time in the volumes of some really key brain regions. So it really is possible to age cognitively and brain-wise in fairly healthy ways. So here's the big question again, what's really different about this group? And it happens to be that they're a little bit older, but they're a little bit less well-educated, which is interesting because they start off at the exact same point as these other groups, but the less well-educated group seems to be changing over time. And I think Christine Yaffe may be talking a little bit about cognitive activity and how that might be something important as we age. So if you really look at multiple studies over time, I think that there are a lot of different ways in which our cognition, our brain structure and brain function changes with age. There's the group up here in red that we all want to be a member of, which is stable functioning over time. You have a couple of groups where you see some subtle changes over time, perhaps not enough to significantly interfere with day-to-day functioning, but certainly the kinds of minor annoyances that we all experience when we forget where we placed our keys a little bit more often than used to. And then there's another group that does seem to be heading down a far more negative trajectory for potentially a lot of reasons. And of course, we're studying folks right here early on, and our goal is to really try and understand or predict at this particular stage of the game which trajectory someone's going to be on. So really, the take-home point is that the way in which brain structure and brain function changes with age is enormously variable. It is not inevitable. Cognitive decline, brain shrinkage, significant brain shrinkage are absolutely not inevitable at all. Many of these age-related declines that we see in brain structure and function could very well be related to disease or some kind of pathology. And the bottom line is that the better we understand the multiple factors that influence how our brains and cognition change over time, the better we're going to be able to do to identify some potential interventions or preventative strategies. And again, this is something that Christine is going to talk about a lot later on. So here's my vision. This is what I think is eminently doable as soon as the science catches up, which is, we're now eligible to go into our physicians for an annual wellness visit. And at that annual wellness visit, you can get a cognitive screen that their physician will know a little bit about your genome. Physician will have access to all of your labs, looking at a whole range of things that might influence the ways in which our brains change. There'll be structural imaging, there'll be functional imaging, there'll be a lot of information on your lifestyle, okay? And then on the basis of this annual wellness visit, your physician can sit down with you and identify some potential risks for you down the road and some ways in which you can mitigate those risks. All right, so that's the vision. So how are we going to get there? Well, again, there's lots of terrific studies going on across the country. This is what we're doing here at the Memory and Aging Center. So to date, we've seen over 850 functionally intact, normal older folks, and by older I mean over the age of 60. Although my definition of older goes up and up every year. We extensively follow about half of that sample, and this extensive follow-up includes a whole range of studies, including structural imaging with MRI, using PET scans to look at how the brain is functioning, or looking at proteins in the brain. We have DNA on everyone. We have labs on everyone. We have CSF on a small handful. And how many folks here in the audience actually have or are currently participating in one of the Memory and Aging Center studies? Okay, great. Thank you, and thank you. We really appreciate your willingness to do all that. All right, so if we're going to try and start teasing apart all the different factors that could potentially influence the way in which our ages brain, it's good to have a working model, all right? And the working model here, which is a little bit on the complicated side, basically says that, this bottom row here is different aspects of cognition. How quickly we process information, how well we're able to multitask, organize, plan, reason, our nonverbal skills, our verbal skills, how well we can learn and hold on to new information. And the basic idea is that all these cognitive skills are really directly related to what's happening in our brain structure and our brain function. So there are certainly different regions of the brain that are particularly important for different aspects of cognitive functioning. And the real big question is what are all the various factors that could potentially impact our brains and our cognition? And I have just a handful of items up here on the top that are candidate topics to review that I'll touch on very briefly, and I know that Saul and Christine will go into a lot more detail on some of these in their presentations. All right, so let's get started. Genetics. What are just some of the different genetic factors that influence how our brains age? The apolipoprotein gene is probably the most widely known one. How many people are familiar with ApoE? It's probably the biggest risk factor for Alzheimer's disease. It comes in a few different flavors, and there's one particular variant of the ApoE gene that can almost quadruple your risk of getting Alzheimer's disease if you have one of those alleles. I'm not going to really talk about ApoE because it's probably the one that gets the most coverage. What I want to do is just sort of mention a gene that you might not be familiar with that I just think is so interesting. And unlike ApoE, which is a gene that increases your risk for something bad happening, I want to say a little bit about the klotho gene. So the klotho gene is something that I think was first really noticed in mouse models. So it's a gene that seems to be associated with longevity. So about 20 to 25% of us have a variant of the klotho gene that's associated with living longer. And it's associated with higher levels of this klotho protein that's circulating in your blood. Right? So there's something about klotho and the klotho protein that is strongly associated with living longer, particularly if you're a mouse. But it's also true with humans. So one of the questions that came up is, well, does this klotho gene also enhance brain structure or brain function? So this is some really wonderful work being done here at UCSF by Jen Yokoyama and Dina Dubal. Jen is a geneticist, and Dina is actually a laboratory scientist who's very interested in measuring blood-related proteins that may influence how our brains age. So they used a few different large cohorts, including the one at the Memory and Aging Center, where we had good imaging data, cognitive data, and of course, genetic data. And the first thing they looked at was, well, is the klotho gene associated with better cognition in these older subjects? And in fact, that is exactly what they found. They looked at a few different aspects of cognitive functioning, and what they found is that the group that was the heterozygous for klotho, these are the 25% that have the advantageous combination of klotho DNA, had better executive functioning. So by executive functioning, I mean they did better on tasks that involved problem solving, novel problem solving, or multitasking. And the difference was really quite significant and was true across the age spectrum. So whether you were a 50-year-old with the good klotho gene or a 90-year-old with the good klotho gene, you did better than your age peers who had the less advantageous klotho allele. There are differences on executive functioning. So what part of the brain is executive functioning most typically associated with? Well, it's associated a lot with the frontal lobes. It's not the only region that really contributes a lot to executive functioning, but it's certainly a particularly important one. So they were very interested to see if the MRI, the brain structure findings, paralleled some of the cognitive findings, and that's exactly what they did. So here's a nice little image of the part of the brain that was actually larger in the individuals who had the advantageous klotho gene. So bigger brains, better cognition. What we don't know yet, but future research is about to tease apart, is whether having the advantageous klotho gene slows cognitive aging at all, or if it simply gives you an advantage from the get-go that you maintain, hopefully, for the rest of your life. So that's the klotho gene. Again, there are lots of other genes that are very important and do influence cognition, but I want to move on and talk a little bit about proteins. Proteins in the brain. Now, I understand that Michael Geschwin introduced the concept of proteins last week. There are a lot of proteins that are circulating that actually do have an influence on cognition. And some of these are really interesting. I mean, estrogen, androgens, testosterone, these all have an influence on how our brains work. But the big ones that I want to talk about are some of the proteins that really can adversely affect cognition. So, everyone's, we've known for over 100 years, say in Alzheimer's disease, that you get these plaques and tangles in the brain that I think Michael may have showed you a picture of last week. But about 30 years ago, a couple of really interesting things happened. So around 30 years ago, they realized that if you really studied the molecular makeup of these plaques, that it was all about an accumulation of abnormally folded amyloid. Amyloid is a protein that serves a normal function in our brains, but basically our cells are always making proteins, breaking them down, making new ones, breaking them down, and at some point this balance gets disrupted and there's an accumulation of these misfolded proteins that start to clump together to form the plaques. Also around 30 years ago, this guy named Stan Prusiner, who's here at UCSF, won the Nobel Prize for raising the hypothesis that abnormal proteins were really the basis for all of the neurodegenerative diseases, not just Alzheimer's disease, but Parkinson's and the frontotemporal disorders. And I think that the last 3 decades are really proving him right. So when you look at some of these proteins, you'll notice that there's quite a few of these proteins. This is a nice picture of a tau protein, and this is how it's supposed to be folded. These are very big molecules that can have all these sort of weird shapes. So it's kind of easy to see how they might misshape and then start clumping together and forming plaques and tangles and other kinds of pathological substrates in our cells. So Alzheimer's disease is associated with amyloid and tau, and Parkinson's is associated with a protein called alpha-synuclein. There's a couple of other proteins associated with different other diseases. So why am I really spending all this time talking about proteins in the brain when I'm really supposed to be talking about normal aging? Why am I talking about Alzheimer's and Parkinson's? Well, about 10 to 15 years ago, a group in Pittsburgh came up with a way of imaging the amount of amyloid, misfolded, clumped up amyloid we have in our brains. It was a great advance because prior to that, the only way you could figure out if someone had amyloid deposits in their brain was on autopsy. So we now have a very sort of fancy imaging process and now there are a couple different ways now to measure amyloid in the brain. And here's what happens when you have Alzheimer's disease. You can see all the sort of the yellow and red regions of the brain that are representing accumulations of this amyloid protein. Okay? We don't want that. It doesn't look good. And here's an age-matched cognitive control. And as you can see, things are looking pretty good, right? This is what we want. This is what we don't want, right? Simple. Alzheimer's, amyloid, normals, not so much, except it gets a lot more complicated and a lot more interesting because these are also cognitively normal, age-matched individuals. And as you can see, this cognitively normal person has as much amyloid in his or her brain as the patient with Alzheimer's disease. Well, that tells us a few things, that, very interesting things. One is that, well, amyloid is not the entire story. Number two, it just raises the question of, I mentioned all these different aging, cognitive aging trajectories, with some groups not holding on to good memory for as long as others. And could it be that the fact that you're normal but have some amyloid in your brain puts you at risk for some declines later on? And this is a topic that still gets debated quite extensively in the literature. And I don't think the answer is all that clear. There's some studies that follow people who are amyloid positive, and they don't really change much at all. And other studies that do purport a higher rate of progression to dementia. So we don't really yet know for sure. But I think that the major implication for understanding normal aging, whatever that is that one of the factors, one of the reasons why you might see some cognitive change in someone who is otherwise perfectly normal, community dwelling over the age of 60 or 70 is that they're starting to have some abnormal protein accumulation in their brain. And in fact, by the time we're in our 80s, probably 30% of us have measurable amyloid in our brains, and it just goes up and up with age. Another really interesting area of research that this opens up is to understand and bottle why people with amyloid in their brains are staying so normal and healthy, because, we would want some of that. All right, so that's the protein story. And again, I think it's a very important factor in understanding cognitive aging trajectories. Health and exposures, I'm not going to say much at all because this is Christine Yaffe's area, but again, I think it's a major player in helping us understand why some people start to change more rapidly than others. Cerebrovascular risk factors, I'm just going to touch on this very lightly because again, Christine Yaffe is going to talk about this. So I think this was shown last week where it's got a nice slice of the brain. The outside of the brain, the cortex is gray, and you have all these white regions, which are fiber tracts. These are the communication lines that link together all the neurons. So it looks white, we call it white matter, and we want to keep our white matter healthy. And here is some abnormal looking white matter on a scan with someone who has a lot of cerebrovascular disease. And these are probably possibly some very tiny vessels that are just being occluded and causing some disruption. Well, if your brain has these sorts of white matter hyperintensities in it, is that a bad thing? And there's still some debate about that, but I think that the overall zeitgeist is you want to keep those cerebrovascular changes down to a minimum. But even in the absence of these documentable signs on imaging, we're seeing that cardiovascular risk factors may have a negative impact on cognition. So a recent paper by Darvis Fraser, who is a postdoctoral fellow in our group, looked at insulin resistance, again, in our completely normal older folks, and looked at executive functioning. And these p-values basically mean that if it's if the number is less than 0.5, it's meaningful. If it's greater than 0.5, we're not going to take it all that seriously. What are the things that predict executive functioning in a large group of normal older individuals? Well, the older you are, the less well you do. But education didn't matter, gender didn't matter, cholesterol didn't matter, body mass index didn't matter in this particular study. Our blood pressure didn't matter so much. White matter hyperintensities did. And even above and beyond all that, your insulin resistance. So those subjects who had higher levels of insulin resistance did less well on cognitive tasks. And a similar finding with triglycerides from Vishnu Parthasarathi, a medical student who just recently submitted this paper, again showing that even after you factor in all these other variables, that just your triglyceride levels potentially has an impact on cognition that may be independent of what your white matter looks like. Inflammation, well, this is Saul's area of expertise. I'm just going to introduce to you the idea that inflammation matters. So inflammation's good. When we have an injury, when we're sick, we have mounted inflammatory response that's very important for healing. And what happens after we get better is the inflammation subsides. But as we get older, what happens is that, number one, we tend to get sick more often. We have more peripheral injuries, more opportunity to have inflammatory disease, and our ability to reduce the inflammation over time is diminished. And we also know that when we have inflammation peripherally, it translates pretty directly into inflammation inside the central nervous system. So a lot of us, with aging, there is a potential for more inflammatory molecules in our central nervous system. A nice little study by Bree Betcher, who was a junior faculty here until recently, wanted to look at the white matter changes that might be associated with higher levels of inflammation. And she looked at several different inflammatory markers in the blood and found that IL-6, the interleukin-6 in particular, was associated with really subtle microscopic changes to the white matter in this particular region of the brain. All right, so let me finish up with one final thought, which is, what can we learn from people who are aging really, really well, super agers? So this is a study just done by Nick Bott.
He's really 90, right?
Yeah, he is. He's amazing. 30 marathons in 30 days. I mean, he says. So one of the things that it tends to be particularly vulnerable to age is information processing speed. We do get a little bit slower as we get older. And then we can measure this and use this to our advantage when we follow folks longitudinally. But there are extremes to how people change in their processing speed, and he wanted to know what we can learn. So he used our longitudinal cohort to identify 3 different groups. So this is information processing speed. So the higher it is, the slower you are. And the benchmark here are people in their 20s and 30s. And so if you have a score close to 1, you're operating kind of like a 20 to 30-year-old. And there's a group of our normal agers who are operating that quickly. And then there's a couple of other groups who are kind of average for their age and their information processing speed. And if you followed them over time, the really fast group stayed really fast. One average group stayed average, and one group actually got significantly slower over the 2 to 3 year period. So he was really interested in what are the differences between these groups. And she didn't see any group differences in things like body mass index, cholesterol, age. But what he did find is that there is a difference in their cardiorespiratory fitness, that the superager groups were just more fit. They had higher levels of exercise, so that's kind of interesting. Their images looked better. They had more intact white matter integrity when you looked at their MRIs, and they had lower levels of inflammation. This is a significant difference on interleukin-6. So I think that, this study of superagers was very illustrative, and I know that Christine and probably Saul will follow up on those findings. So to summarize, aging, can be associated with changes in brain structure and function, but these changes are not inevitable, and they vary quite considerably across individuals. There are so many factors that influence these rates of change, many of which are remediable or addressable, which gives us some power or influence on how our brains change over time. So with that, I thank you, and I'm going to pass the baton to Saul. Because of the filming, we're going to wait until after we're all done before we do questions. Thank you very much.
Great. So my name is Saul Villeda, and I'm relatively still new faculty here at UCSF. I'm starting my 4th year here. I'm over at the Parnassus campus. And today I want to share with you a bit of basic science, what we're currently learning about aging and cognitive aging still from our animal models, and what that might mean for future translational approaches and therapeutics. So I want to present to you sort of a systemic approach for rejuvenating the aging brain. Now, the question I have for you, and I want everybody to raise their hands, how many of you think that the effects of aging can actually be reversed? Not prevented, but once we're already old, we can actually go backwards. How many of you believe this? Okay, not too bad. So this is sort of the main question that drives my research, my interests, as well as my lab. I'm a neuroscientist by training, so we get a little bit more specific. And the question I'm asking is, can the effects of aging on cognitive function be reversed? So again, once we've already gone through our entire lifespan, I like to think that I'm still here. People are starting to say I'm going more towards here. I still have a ways to go there. And the idea would be, once we are here, how do you get back to what I feel like and how I act now in my early and mid-30s? So that would be the idea. Now, as was mentioned, sort of one of the things we think about is that group that is declining with age, right? That's the group we're really interested in sort of targeting. And we all have a certain level of lifespan. It actually hasn't changed at all. The maximum lifespan is still between 110 and 120 years. It has been for a really, really long time. And in my lab, we're not interested in changing that lifespan, but we are interested in changing our health span. Why? Well, as we get older, not only are we sort of having some cognitive impairments and a few nuisances that happen with it, the susceptibility and vulnerability of our aging brain to age-related diseases is actually increasing. So even though we may not see it, a lot of us are having changes in our brain that are going to allow those diseases to happen. Now, what happens if I don't change lifespan? He still stays there, but I actually increase health span. What if I can keep or bring your brain back to a younger state? Now, imagine if we can move both of these charts over. We're still having the same endpoint, but maybe rather than having this many people with neurodegenerative diseases, we're actually only going to have this many people with neurodegenerative diseases. So the idea of thinking about health span, thinking about aging as something that we can target before the onset of disease, is starting to really become a really exciting sort of therapeutic consideration now. And that's one of the big drivers of my lab. So I don't do human research, I do animal research, and I'm going to try and show you why I think these guys are still good models for us to understand a lot about humans. So let's put this into everyday context, this idea of age-related decline. So I drive from Menlo Park into San Francisco every day. We're going to pretend that I can actually afford to buy a hypothetical Tesla, okay? So just run with me on this one, okay? So, of course, Yeah, and of course, this is a garage apartment at UCSF where everyone has Teslas. So I'm driving in the morning, and I'm gonna park my Tesla, hopefully someday, in an area, and I have to remember after many, many hours, I'm a hard worker, let's say 16 hours, I come back and I have to remember where did I park my car? And my brain is gonna sort of use certain things. So we're in San Francisco, so let's say at Parnassus, I actually do get to see the Golden Gate. Usually, let's say there's some sort of rainbow flag, and this is actually much more popular on the Parnassus campus, which is food trucks. They have really good food trucks. So let's say from this parking lot, I can actually see these sort of scenes. They're gonna serve as visual cues. So I'm gonna go to work, come back, and my brain is just gonna pick this up and go, ooh, bridge, flag, taco truck. And it's gonna guide me, right, to where I actually parked my Tesla. So that's a form of learning and memory. It's called spatial learning and memory. As we get older, this becomes a little harder. And sometimes, rather than using these spatial cues, you'll see people with their car alarms kind of running around the parking lot, trying to find their hopefully not hypothetical Tesla like mine. So what's interesting is this same type of age-related cognitive impairment is actually observed across species in animals. So think about it. There's a parking lot and there's some spatial cues. Well, guess what? I can use animal models to have the same exact type of learning and memory, and now I can actually do experiments. Just like a bridge, I can use geometric shapes around a pool of water and study learning and memory, spatial learning and memory in mouse models. And that's what my lab does. So what we do is we put a little mouse in a pool of clouded water, and there's gonna be a hidden platform. And just like my brain's gonna recognize the bridge and the flag and the taco truck, it's going to recognize these spatial cues. We put them in there a few times, and a young animal, they learn so quickly. They hate cold water. They're gonna learn how to get out of that so fast. So again, using these spatial cues. Now, what happens with aging? Well, he can still find it. That doesn't change. But he does have a much harder time getting there. So he takes longer. He doesn't quite have as nice of a sharp turn, but he does get there. So what's great about these models is it allows us to study what is promoting these aging effects in a young animal, and maybe even how can we take an old animal that's already old and bring it back more towards a more youthful state. So spatial learning and memory in these mice is governed by the same region as in humans, known as the hippocampus. So here's a region. This is a mouse brain that was just cut coronally, so it was just cut this way, and I'm zooming in on the hippocampus. Ours is, of course, prettier and more complex, but it serves the same purpose. I'm gonna zoom in on this one region, okay? And the reason this is so important is it's actually the most vulnerable region to the effects of aging. It's actually the first one to start showing some impairments and some changes in the way that it looks. So let's zoom in. This is again the hippocampus, but now I'm gonna show it to you in terms of how it looks like in its structure. And the brain is just a bunch of circuits, it's a bunch of electrical activity that's signaling to each other through these nerve cells, through these neurons, and that's how all processing is formed. So this particular region is considered a trisynaptic circuit. There's 3 pieces to this circuit. We're gonna get all this information coming in from our environment, the bridge and the flag and the taco truck, and it's gonna come in and it has to get filtered. And we have to decide whether we wanna keep it as a memory and it's important, or we can just toss it. So that information is gonna come to that first part of our little circuit. And it's gonna come here to what's known as a dentate gyrus, or DG. Now this is a really, really, I think, cool structure because it's our filter. It's that first place where we make that decision between keeping and letting it go. Once that decision is made, it's gonna get filtered throughout and processed throughout the rest of the circuit, CA1, CA3. Then the nice packaged information that's been going on through these nerve cells, these neurons, goes back up to the higher regions of our brain, and it's stored there. And then when you come back from work, it'll guide you, right, based on those cues. This is a really unique and dynamic region. That DG I told you about, it's actually one of the few regions that still has stem cells in it. So we can actually still produce new neurons even when we're older. And those are really, really important for strengthening some of these memories. The other thing we've now learned is that the immune cells are actually also really important in this circuit. And they're actually a good thing. In a young environment, we want really happy, active immune cells because they're gonna be surveying, they're really dynamic. They put their arms, they stretch them out and in, and they're really checking to make sure that these connections are nice and tight and that this region allows for new neurons to be produced. And then age happens. And then this happens. So you'll see that my nice stem cells are they're more like dormant. They're not completely gone, but they're mostly asleep, and it's really, really hard to get them out of hibernation. There's a lot more immune cells, and they're much angrier. So they're active. There's inflammation now occurring in the brain. However, until you get disease, there's actually no cell death. However, what happens is all of these changes collectively are going to have a really big effect on where the neurons talk to each other, known as the synapse. And these little spines that you see here, it's just a zoom in of where each one of these is one of these connections. Every single one of these spines is a point where this neuron is talking to other neurons. We have thousands and thousands and thousands of them. However, what ends up happening with age is that we lose the dynamic ability of these little spots of contact to maintain communication. So really our communication is weakened and we get functional decline. And it's not until we develop the disease that we actually see cell death. So we really like taking this from a systemic point of view. We think of aging, it's not just the brain, it's the whole body. And it must be affecting the entire organism. And we thought, what is connecting all the organs in our body together? How do they communicate with one another? And the idea that we came up with, and this was during my training at Stanford, was blood. Blood connects everything. Well, in a mouse model, how can I mix blood together? How can I, change them and make young and old blood happen? We used a really old surgical technique, first used in the 1600s, okay? It's known as parabiosis. And what we do is we physically connect two animals so that two hearts are pumping one bloodstream. What this lets us do, they're quite happy and they can live their whole lives this way. It's like a buddy for life, okay? Think buddy for life. Of course, we take all the preventative care, but really, if we actually separate them, just so you know, and we take them away from each other, they show stress signs. They really get used to having that other animal next to them, so we actually have to keep them together even if we do separate them. So they really do get used to it. But this is a really, really powerful model because it allows us to ask, what is the effect of the old blood on the young animal, in particular the brain, and what is the effect of the young blood on the old animal? And that really can give us two different questions and two different approaches. One is, are there pro-aging factors in our old blood accumulating, and maybe we can target them? And then are we losing some pro-youthful factors? Maybe we just bring them back up and we can maintain a more youthful environment. So during my training prior to UCSF, we actually discovered that if you look at a young animal and then expose it to old blood, those same hallmarks of aging, more immune cells, less stem cells, we could actually mimic that. So there was something active about old blood that was promoting aging type effects. When we looked at young animals that were exposed to old blood, we saw that same impairment. Now, don't think that I'm putting these two guys into a pool of water. I'm using this as a representation. We actually gave young animals old blood intravenously, okay? But we saw something like this. It was very similar. Not quite as bad as old animals, but very close. So the first question we've asked in my lab was, okay, well, can we decrease some of these blood factors from old blood, and does that cause improvement in cognition? But in order to do that, we first have to figure out what are potential factors in old blood in the first place. And what we did is we took a nice cohort of old animals between 6, 12, 18, and 24 months to put this into sort of human relevance. A 3-month-old animal is approximately someone in their 20s. And in between 18 and 24, you're sort of in your mid-60s to early 70s, just to put this into context. You start getting those cognitive impairments at 18, which would be in your 60s. So what we did is we took the brain of these animals, and then we also took the blood. And we asked a question. We asked, if we look at a few factors, can we find factors that are changing in a way that's similar or correlating with the changes we see in the brain? And I told you a few hallmarks. I told you stem cells change. So we looked at these stem cells. This is actually a section of a mouse brain, and they're stained just for a protein. It's known as double corn, but all this is, these little guys are newly born neurons. And we asked, how did their level of sort of new neurons, how does that correlate with changes in immune factors, or changes in blood factors, sorry. And then we said, okay, we got a number of factors and we wanted less. There was too many of them. And we said, okay, let's look at another cellular hallmark. Let's look at those immune cells. And those little dots right here are just labeled immune cells in a young brain here. They're pretty happy in a young brain. So then after all this analysis and looking at all these factors that were changing in the old blood, we looked at about 70 of them. It turned out that we did identify a number of factors. And one of the ones that my lab has been focusing on is called B2M. And this is actually an immune molecule. It turned out we found a lot of immune molecules. It seems that systemic inflammation correlates really well with age-related changes in the brain. So inflammation seems to have a very important role. So what on earth is this, right? It changes with inflammation, it changes with stem cells, it even changed in our parabiosis model. Everything we had looked at, this thing is changing. So what is it? Well, it's a really interesting molecule. All of you, I'm sure, have heard about tissue rejection. When someone needs to have an engraftment, a lot of times we're worried about the tissue being rejected. Well, it turns out that this molecule is really important in that tissue rejection. It works along with other molecules. They're called MHC1. All that matters is that they're the ones responsible for telling your body, you are part of me and I like you, or you're not part of me, I'm getting rid of you. And that right there, that simple concept, is what causes tissue rejection. So one role in which it has is an immune role. There's all these cells, immune cells, all in our blood, and these molecules are the ones signaling to these guys, come, rescue me, this is foreign. Maybe if you have a viral infection, this is what's gonna tell your body, this cell is infected, get rid of it. So that's one role, the immune role. Turns out this is a really clever molecule because it also has a completely different role in the brain. Turns out, same molecule, and this is that connection that I was telling you about, those little spines that I showed you in that one black and white picture. Turns out this guy's really important there too. It's telling the brain, you have too many connections. We don't need this many. Get rid of some. This is really important for things like your visual system. When you're born, way too many connections, and you have to start getting this input, visual input, and it starts sort of refining right eye, left input, left eye, right input. Same molecule, an immune role and a brain role. And we thought, this is really cool. You start looking at the human literature, and it turns out this guy keeps popping up. People with HIV-related dementia, this B2M actually goes up really high in their blood and cerebral spinal fluid. Alzheimer's disease patients also have much higher levels in CSF. So we thought, interesting. It's been implicated in cognition, it's immune, it's brain. Let's look at it. So that's what we did. So the first thing we did was we just looked at plasma levels, just the blood without the cells, just basically the juice. And we said, how much of it is it? And as we go up with age, there's an increase in this immune molecule. We looked in our parabiosis animals, so the young attached to the young with the old, we see an increase. Then we thought, what about humans? Is this also changing in humans? We got samples from just normally aged humans between the age of about 24 to about 96, and it also goes up in the blood of humans. And in a more modest number, but we were able to get cerebral spinal fluid samples, and look at that, between 20 and 45, low levels. After 65 to 90, you'll see that there's much higher levels. So not only are we seeing this in our mouse model, we're actually seeing it in normal humans. So as we get older, this thing is going up in our blood. So now we use mouse models for a reason. We wanted to start getting functional. We wanted to start tweaking things and asking what happens. So we said, okay, what happens if we take a young animal and make it look more like an old animal by just increasing the blood levels of this protein? And that's what we did. And we just injected it into the mouse. And then we did cognitive behavior. We just behavior. We wanted to test them. And today I'll just share a little bit of data from our first one, our water maze that I talked about before. So the first thing we did is first we looked in the brain. And you'll remember this stain that I showed you, right? This is just stem cells that are producing new neurons. Young animal, just receiving normal saline, has nice levels of stem cells there. Now, after we added this protein, it starts looking more like an older animal, at least in the brain. And then we thought, what about learning and memory? Now, we use that same water maze, but we make one change. We try and make it a little bit tougher. We actually divided it into arms, and we said there's still, the triangle, and we still have the rectangle and the stripes, and there's still a hidden platform, but this time we're asking, how many times do you go into one of these arms? How many times do you get confused before you find it? And that's what we did. We tested them. And what happens? On that first day, we asked, how many times do you go into one of those arms that's not the right one? How many mistakes do you make? And they can still learn. They start at 4 and they end at about 2, so they still learn. And then that second day, when we test them again for how well they remember, These young guys that have just saline, wow, they make less than 1 error on average. These guys are just jetting into that arm with the platform. We add this protein, this immune molecule, they're doing quite worse. Not as bad as an old animal, but way worse. So then we started asking, what about if we totally take this away? Let's just take it away from the body, from the blood, from the brain. Let's get rid of this immune molecule. One of the powerful things about using these mice is the fact that we have genetic tricks that we can use with them. And that's just what we did. We used one of these mice that doesn't have this protein, this immune molecule at all. And we said, what happens when we look at a young animal? And then what happens when we look at an old animal? Are there any differences between them? And this was all work by my graduate students. So first they looked at those stem cells again, and they saw no difference. You take this protein away in a young 3-month-old animal, totally fine. They were actually, my students were quite sad about this because they spent a lot of time, there was no difference. So I convinced them and I said, you know what? There's no difference at 3 months. Why don't we wait another 1.5 years and see if there's a difference in an old animal? And somehow I convinced them to do this, and this is what we saw. There's actually an increase in the number of these new stem cells and these new neurons. Now, even though there's not that many, one stem cell can have hundreds of thousands of connections when it becomes a new neuron. So even a few hundred more can actually have a huge effect. We also think, of course, other things are happening in the brain, but this is just one that we looked at. What about behavior? Well, you'll see here a young animal, they start making about four errors and they go down, and then on the second day they learn much better, they get to about 2. However, when you look at this old animal, one of the smartest old mice we've ever tested in my lab, I mean, you get rid of this thing and they're learning down to about 1.5 errors, which is so much better than they would normally at 3 and 4 errors. So it seems to be age dependent. You get rid of it when you're young. It doesn't seem to be that important, at least for the brain, for learning and memory. It really seems to be that something happens as you're getting older and you have that bump in it that is what's causing some of this impairment. So it's really great because it starts giving us a handle, at least in thinking about therapeutics, things that we can target in the blood that could have an effect in the brain. So to come back to this question, can decreasing these pro-aging factors have an effect? Well, I've given you some tantalizing evidence that it might be possible. However, the home run would be making a drug, an antibody, a small molecule to sequester or block this guy in a young animal and then go, can we actually reverse it? So we're able to prevent the decline. Are we able to reverse the decline? So in keeping that idea of reversibility, I'm going to give you a short little story about a way that we may also be able to. So this is one approach. What about the opposite? How about the young blood? Can that do something? Well, it turns out when I was a graduate student doing research, there had been this body of literature in other tissues, muscle, liver, that showed that young blood can actually enhance stem cell function in different tissues. So during my PhD, I looked in the brain, and even though there's this blood-brain barrier, it seems that it's still important, and it can enhance that newly born neurons. More neurons are produced in an old animal if you give them young blood. So then I wanted the sort of home run. So right as I was leaving my postdoc training and then starting my lab here, so part of it I did there and part of it my students did here. So we've seen this effect that I'm going to show you, 2 institutions. We saw it at Stanford and we saw it at UCSF. So I really believe in what I'm about to tell you. So what we said is, can the beneficial effects go beyond just stem cells? I'm a scientist and I really believe, we were talking a little bit about genomics and we're all really interested in personalized medicine and how our genes are going to be able to really help us sort of, customize maybe our therapies. Well, we took advantage of genetics. We took advantage of genomics. And I said, okay, what if we take the brain of this guy after it's had young blood? Just look at it. What do the genes tell us? Now, this colorful graph here, all you need to know is that some of the genes go up, some of the genes go down, but these genes are sort of their own calling card, and they're able to tell us what certain processes are changing in the brain. They have sort of like their own address, and this right here is the address for synaptic plasticity, those connections that I was mentioning to you that go away with age. We actually found a signature, the address of the same process in that old animal after exposure to young blood. What's cool about that is that's the reason we have learning and memory. That's the reason we have impairments and we have improvements in learning and memory. So we got really excited. And then we went and we looked at the actual connections. Again, an old animal after young blood. Here you'll see not that many of those dendritic spines, those connections between the neurons, but look what happens when you give it young blood through parabiosis. You're taking a neuron that's been there forever. It's been there 18 months, the equivalent of 60 to 65 years in a human, and we're able to bring back at a structural level some of its youthfulness. So what about cognition? And again, this we did, like I said, multiple institutions, tons of times, because we wanted to make sure if we saw something, it was right. So we gave it young blood, just 8 injections over 1 month, about 5% of their volume, and then we tested them. And you'll see here on the first day, they learned just well. There's already a separation. The red ones are young blood, and then the gray ones are old blood. And you'll see that next day, these are normal old ones. They're still making about 3 errors. These guys are acting much more like young animals. So even though they went 18 months and they went through the entire aging process, just giving them some young blood was enough to bring this back and make them more youthful, even at a cognitive level. So to come back to this sort of overall question that I had, what about reversing it? So right now my lab's been really interested in understanding how, and that's something that's ongoing in my lab. But we really, really believe in this idea that there is a certain amount of reversibility to some of these effects of aging, even in the brain, even at a cognitive level. So not only is preventative care one way of approaching it, but maybe reversibility is also a way of approaching it. And this is all stuff that's ongoing. There's a great sort of environment to do this because there's so many crosstalks that we're having with clinicians and basic scientists. But the end result, I think, that we're all getting from studies like Clotho to studies like Young Blood is it doesn't have to stay static. Aging does not have to stay aged. We can actually bring it back. And I think that's really exciting. Just so you guys know, some of the things we're doing right now, we're going back into that genomics. We're so interested in understanding what is it about an old brain that we're actually even tapping into. I mean, something is left in there that's just asleep, that we're waking up with using young blood. What about cognition? I showed you one type of learning. What about other types? What else can we rejuvenate? What are the limits on it? Is there such a thing as too old? Or can we constantly bring it back? Do you need exposure to young blood forever? Or is one dose enough to keep you going for a while? Those cells I told you about, right? The stem cells, the neurons, the immune cells, what's their contribution? Do we have to fix the whole brain to make it younger or not? And of course, just functionally, other parts of the brain. Is it just the hippocampus, or can we do this throughout the whole brain? So this is, again, some of the ongoing research that we're doing in these mouse models that we hope one day will actually be able to translate into humans. And just to give a little shout out, these are some of my graduate students. This is my entire lab. And these 3 had a lot of work to do on the immune as well as the young blood. And actually, one of my master's students is now getting his PhD at Davis, who did a lot of the young blood work as well. Thank you.
Thank you for persevering. I'm the last of the 3. So I'm going to talk a little bit about try and tie together a little bit what you've heard so far, and really focus in on what are some things that we can do short of being fused with one of our litter mates. What else can we do that might have a good effect in terms of cognitive aging and prevention of dementia. So that's what I'm going to really focus on. And Dr. Kramer mentioned I was part of this Institute of Medicine report that we just finished a few months ago, and a lot of what I'm going to talk about sort of jives with what we ended up saying in that report, and I'd be happy to point you in that direction as well of our report. Okay, so why is this important? Now I'm going to talk a lot about dementia, and I think Dr. Geschwin last week introduced the concept of dementia. When we're talking about dementia, that's the syndrome. It's not the cause of dementia. It's not Alzheimer's per se, or vascular, or FTD, but it's just the syndrome of having cognitive decline, usually with aging, that's enough to be affecting your functioning. And we know that this is a big problem because, a lot of people have it. It tends to go up with age. But because of the demographic shifts, because of 2 things. One, we're living to be older. Our life expectancy is much higher than it was 50 years ago. Now, in fact, the oldest demographic of our population is the 85 plus. So the 85 plus group is really going up exponentially. So we have people living to be longer, older. And we also have in this country and in many European countries and in Asia a shift with the baby boomers aging. So we have less less children being born, and the proportion of our population that's aging is different. So instead of having a triangle like this in terms of population, it's becoming sort of more like this, or even inverted. Because of that, we are going to have a dementia epidemic, really. And I can't overemphasize this really enough, because I think in the U.S. we're just starting to wake up to this, and you can imagine there's a lot of policy and politics, and we have to think a lot about our healthcare. We have to think about research. We have to think about, certainly the funding of all this. It's a huge issue, and I think we're a little behind. We are going to have, in the next 35 years, a tripling of dementia. So this is a huge issue. And we all have to, I think, really, get a little bit more sober about this, because we are all going to be affected by dementia. And there are a lot of issues around who's going to take care of patients, the cost, et cetera. So we really want to do something about this. And it turns out that because this is so common, because dementia is so common, if you could delay dementia, or even if you could prevent it, that would be lovely. But even if you could delay it by a year or two, you would have a big downstream effect. So this graph shows you that even if you could just delay dementia by a couple years, the downstream effect in terms of a public health perspective is going to be quite large, and the numbers would be quite significant. And so that's what we're all quite interested in. Ideally, we're going to have much better drugs. Right now, our drugs are not very good. They help with the disease a little bit in terms of Alzheimer's and some of the other dementias. They improve the symptoms a little bit, but they don't really change the slope of the disease. They don't really modify what's happening. And we don't have anything really for prevention. So this is really what we're trying to get. We're trying to get something that will change the disease course, but ideally even prevent. And there are a lot of studies trying to tackle this that I'm not going to talk so much about. I still think we're 5 or 10 years out in terms of some of those. But you get the concept. What we really want to do is ultimately have better treatments, but ideally do something to prevent or delay. So I'm going to talk a little bit about what we can do now, because the drugs are in development. There have been some frustrating leads that haven't panned out. What can we do now? And I'm going to focus really on lifestyle factors, primarily lifestyle and health factors. These are the things that we can do now. And I think it's really important, because we keep hearing, it's going to be another 5 or 10 years. It's another 5 or 10 years. But, we can't just sit around. We have to, I think, look at some of the things that are very promising. And these are the things that I think we can do now. And I'm gonna show you some of the evidence why we think these things are really important. And then I'm gonna show you, if we actually deal with these things, what the implications might be. So number one, take care of your heart. And Dr. Kramer alluded to that a little bit when he talked about some of the work with the pre-diabetes and some of the other things. I'm going to talk a little bit more about that. So what we mean is that the things that we think about as heart disease factors, high blood pressure, diabetes, cholesterol, these are the things that actually also affect the brain. And we think that these are one of the best strategies. Be physically and mentally active. I'm going to talk a bit more about that. And again, Dr. Kramer mentioned the fitness issue, and also mental stimulation. We're going to talk a bit about that, because these we think are very important. And they're not that costly. There's no side effects. So they're great. And then I'm going to talk a little bit about sleep, and I'm just going to mention depression, although not go into it so much. These, I think, are also really, really important. Sleep in particular is getting a lot of interest now as something that we think is very much affected and may correlate with dementia risk. Okay, so just a little bit on cardiovascular risk factors. Again, it makes a lot of sense, right? The same blood vessels that are affected by high cholesterol or diabetes or high blood pressure, the same blood vessels that are in the body, they're in the brain. So the same things that give you atherosclerosis in the body could give you atherosclerosis in the brain. And you might see it on the MRI that Dr. Kramer showed, it mostly with the white matter changes. That's what we often will see. So this is some work we did a long time ago, although not 25 years ago, because I wasn't even in San Francisco 25 years ago. So don't believe anything he says. Anyway, we looked at this issue and looking at different cardiovascular risk factors, and many, many people have now done similar studies. I'm just going to make a point that we think it's stronger if it's in midlife versus late life. And what do I mean by that? What I mean is if you have high cholesterol or high blood pressure or diabetes, there's very, very clear an association in your 40s, 50s, 60s, and developing dementia. Later on, it gets a little trickier. So if you start looking at people in their 70s, 80s, 90s, the association's less clear. And I'm spending some time on this because it's really important. What you don't want to do is have somebody who's 90 and be super aggressive with their diabetes or their high blood pressure. You might cause more harm than good. So I just want to point that out, that a lot of what we're talking about really has a lot to do with sort of earlier in life, midlife, early, late life, but less much later. And I think it's an area we need to understand a little bit better about. What does that mean? Why is that? And when is the right time to intervene? Okay, I mentioned this idea of use it or lose it. People say that a lot, use it or lose it. What do they mean? They mean the brain and the body. You've heard from Dr. Villeda a lot about some of our rodent models and what they can teach us. And I think that this study, which is now actually quite dated from 1999, I think it's what was one of the most interesting studies I ever saw, because what it showed us was that if you took a rodent model who was sort of your couch potato, who didn't do that much and had sort of your boring cage, sat around a lot, that they didn't have so many new neurons developing. But if you took a mouse that was a runner, exercising a lot, or one that was in an enriched environment, they had a lot of new objects, new smells, new things to touch, they were all busy learning things. What you find is that there were new neurons, many more, in the brain, in particular in the hippocampus. To me, this is so interesting and so exciting because it tells us that the brain can change and grow. And it's true in humans too. We used to be told, and we used to be taught this in medical school, you're born with all your neurons, and every time you hit your head or you have a glass of wine, you destroy some of your neurons, right? That's not the case anymore. And it's quite fascinating. It's just not the way it is. You can develop new neurons, new connections at every age, and including in late life. So this has really renewed our concepts of what we can do in terms of all kinds of therapies. And it tells us that the brain is plastic, if you will, plastic in the sense of changing and growing. And this has such wonderful implications. It tells us that we can do something about it, and we can try and buffer the brain, make those new connections, so that maybe we can protect what's coming down the pike in terms of all those proteins that Dr. Kramer showed you. So how can we do that? How can we buff up our brain? How can we build up our brain so that there are better connections? One of the best ways, believe it or not, is physical activity. Not only is it buffing up your body, but it's buffing up your brain. And we think that's the case because it actually stimulates new neurons growing. It improves your blood flow. It decreases inflammation. There's all kinds of other things. It decreases your atherosclerosis. This is a study we did actually a while ago, and we just looked. I actually didn't believe it at the time. I thought this is too good to be true. This is one of the first studies actually to ever look at physical activity and cognition in older. This is in older women, and these were women who walked. This was not doing a marathon or anything fancy. These were just, we said to these women in the study, how much do you walk? we broke it down into block equivalents. And it turns out the more you walk in a very nice kind of pattern, the more you walk, but it didn't have to be a huge amount. Even walking a mile a day was actually quite beneficial. So I think this is really exciting. And it tells us that physical activity, and many studies now have shown that being physically active actually prevents cognitive decline and actually changes your hippocampus. You can look at trials, and they show that the hippocampus is bigger, probably related to the new neurons growing and changing. So I think physical activity is really exciting, and most studies, not all studies, but most studies really suggest that this is a way to go. Oops. What about, I mentioned buffing up your body, buffing up your brain. So we think that you can actually buff up your brain. And again, a lot of studies have said if you start with higher education, higher IQ, if you've been involved in professional activities that were more complex, if you do leisure activities that are more stimulating, these, and not sitting around watching soap operas, that we think all of these things actually continue to make the brain have connections and have this buffer, so that if you do start to see some proteins, you have the ability to withstand that. And I think that's one of the real questions. Dr. Kramer showed the different brains with the different amounts of amyloid. Well, it may be that those who have more of this buffer, even if they have some amyloid in the brain, they're not showing the symptoms because they've almost built up a resilience, if you will. It may not be 100% resilience, but it seems to be a buffer. And we call this cognitive reserve, this idea that you can have some reserve to fend off some of these insults. The reserve may be because of your early education, your early quality of education. It may be because of late life activities. So I think a very, very important concept. Now, many of you have heard about luminosity or all these different brain games. And I think this is one of the things that everybody wants to know. Can I do these brain games, and can I build up my brain to protect this kind of possible protein accumulation? This was a trial that some colleagues did, and what they did was they took normal people, these were normal elders, they didn't have dementia, and they trained them on different activities. They trained them on memory, reasoning, and speed, the processing speed that Dr. Kramer mentioned. And sure enough, after just a couple months of training, people got much better. They got much better on learning how to do those things. And it lasted quite a while. This was after a year, but then they went and they looked at it, and it lasted for a couple years. So we know that you can get better at things. If you're worried about your memory, you can actually try and train yourself on memory, and you will get better on memory. The question that everybody wants to know is, does it extend beyond the thing you learned about? So if you do these kind of trainings, does it go to beyond the thing you just learned on? And that's the thing we don't really know. Okay, so there's no question that you can train your brain and to get better, but we don't know whether it then will help with everything and help protect against dementia. Those studies really haven't been done. And you have to be careful because a lot of the advertising is pretty slick. And you have to really dig deeply. That was one of the things we struggled the most on our report, on our IOM report. It had the most controversy because it's a big business. And so, you have to be careful. I can't say whether it's worth investing in a particular type of software or not. All I can tell you is the concept is quite important. This concept of reserve, I think, really does make sense. And you can get better if you train your brain. Whether it extends beyond that, we don't know. Some of Adam Ghazali and his group here at UCSF, they've done some work showing that maybe it does extend beyond, that if you look at certain things, if you're using these certain video games which are teaching you how to multitask, that may extend beyond. But I think that the data is still actually too early to tell. And not many people have really looked at this. So again, I would say this to me is one of the most exciting concepts, the idea that you can change your brain, make it healthier, more synapses, more connections, just like maybe in the young mice. But be careful, caveat emptor. We still don't know a lot, and we can't really recommend any particular kind of stimulation per se, but a very, very important area for future investigation. What about sleep? I mentioned this, and I'm going to go through this quickly and then sort of trying to sum it all up. What does this all mean? I think, again, this has been fascinating. It turns out that if you take mice and you sleep deprive them, okay, you wake them up a lot or you don't let them sleep, it turns out they have a lot more of the amyloid protein building up in their brain. We think that we still don't know, what does sleep do? But it must do something very important because we all have it, and it's preserved in all the different species. And we're just starting to get a sense that probably one of the most important roles of sleep is to clear out the sort of the metabolites and the byproducts that have accumulated in the day. And so you need sleep as sort of a downtime to do some house cleaning, to clean up some of the debris. If you don't have it, some of that may build up and you might have some of the abnormal proteins that Dr. Kramer mentioned. And indeed, we and others have started to really look at this. We have found that sleep-disordered breathing, otherwise people think of this as sleep apnea, that if people have sleep apnea, they actually have an increased risk of getting dementia. And we think a lot of it is because they're not getting enough oxygen to the brain. What happens when you have sleep apnea? You stop breathing, then you wake up, you stop breathing, wake up, hundreds of times during the night. And that we think it's the low oxygen that may actually be causing some damage in the brain. We also have looked in some other studies showing how much people are efficiently sleeping. So when they're in bed, how much of the time are they actually sleeping versus awake or tossing and turning? Or how long does it take them to go to sleep? And in both of those cases, it really looks like it has to do with their increased risk of developing dementia and mild cognitive impairment. It doesn't seem to matter so much how much you sleep, your total sleep time, because we're all different. There are people who only need 4 or 5 hours and they're fine. And then there are people who need 10 hours. It's not so much about how much you sleep, it's the quality of how you sleep. Okay, so now what I want to do, as I talked about, I want to try and put this together. So what we did a few years ago is we said, all right, let's take the things that we think we have the most evidence for, that if you could modify them, they decrease your risk of dementia. So we looked at these 7 things, most of which I've talked about. Diabetes, being obese, which I didn't really get at, but has a lot of overlap with cardiovascular disease and with diabetes. We looked at physical inactivity, we looked at smoking, we looked at hypertension, depression, and low education. Okay, we looked at all these things and we said, okay, how much of these things are really contributing to the risk of dementia? And what if we could actually modify them a little bit? And so it was sort of like a thought experiment. And what we said is, okay, if you just change these activities, if everybody smoked 10% less, or if everybody exercised 10% more, or even 20% in yellow. We didn't say you had to do 100% change, but if everybody changed a little bit, then actually, again, the downstream effect of how much dementia there would be enormous. You could make a huge difference in how much dementia there was in the world if you could modify these 7 things just a little bit. So I think it's really, really exciting. And you would say to yourself, okay, what's happening in the real world? Well, what's happening in the real world is really interesting, and it's something that you don't hear that much about. And you don't hear much about it. It's a little confusing because I told you my very first slide was we're going to have a dementia epidemic, right? We know that because of the life expectancy and the demographic shifts. We are definitely going to be seeing much, much more dementia. But what a number of studies have now done is they've taken within their study, they've looked at 2 points in time. They looked sort of in the 90s and then maybe in the 2010 or 2000 and about 10 or 20 years apart. And they looked using the same methods. They looked to see how much dementia was there in their study. And lo and behold, what they found now there are actually about 7 of these studies. I showed you just 4 here, but across the world in about 7 different studies, what they're finding is, contrary to expectation, dementia was actually plateauing or going down, which is sort of remarkable. And what we think is what's going on is that this is the same time when we had better care of cardiovascular disease. So statins were introduced. There was better education in terms of some of the secular trends. So it's really interesting that we've seen that by modifying some of cardiovascular disease care, having better education in schools, a lot of these countries, it was post-World War II, there were big changes in nutrition, in socioeconomic status, and education, particularly for women. So we think that actually we can have a big public health effect by improving in these ways. The problem, of course, is that at the same time, we're dealing with a change in lifestyle. People are more sedentary. They're sitting around more in their jobs and in their leisure activities. There's much higher rates of obesity and diabetes. So I don't know what's going to happen. I mean, from a public health point of view, we're sort of looking at shifting tides, and I think it's going to be very important to see how this turns out. Last slide I want to show you has to do with a recent study in Finland, where what they did was they took the things that I told you about, physical activity, cognitive stimulation, cardiovascular health, and they did a trial for 2 years. They said, okay, we're going to take half the people, and we're going to really try and maximize how they do with their heart disease and with their physical activity and diet and all these things. And then they looked in a control group, and they didn't make those changes. And lo and behold, they had pretty big differences in the cognitive change over those 2 years. So it's very exciting. It tells us that actually by modifying these lifestyle factors, you can actually slow the decline, slow the slope, decrease the decline in slope, and maybe even prevent dementia from some of those other studies. So I think it's very exciting. At this point, this is my last slide, I just want to say that, this issue of how we prevent dementia, how we can make our slopes more like Dr. Kramer showed in the flat group or even the improving group versus the decline group is something everybody wants. And there's a huge public health angle here in terms of our whole healthcare system and economy and caregiver burden. So at this point, there really are a couple things that I recommend to all my patients and to everybody, and that's cardiovascular disease management. What's good for the heart is good for the brain. I didn't talk a lot about depression, but it's a very interesting topic. Make sure people are paying attention to depression and getting treated for it. Being physically active, doing cognitive stimulating activities. You don't have to play bridge if you don't like bridge. Do things that you like to do, but feel that your brain is learning new things and making those new connections. And pay attention to your sleep. If you're concerned about sleep, talk to your doctor, maybe be evaluated by a sleep specialist. We need a lot more studies. And ultimately, of course, my prediction is that dementia and cognitive health is going to be like heart disease. It's a combination approach. We're going to have medicines, probably more than one kind of medicine. We might have, like blood pressure medicine and statins and aspirin, and there'll be lifestyle factors. You might have to change your diet or exercise, etc. So I think that we're going to take care of dementia in 10 years the way we're taking care of the heart. And I think that's going to be a much more healthier way to go. Thank you very much.
So the question had to do with the slide earlier about amyloid, and was I saying that amyloid doesn't matter? Oh, I think amyloid matters a lot. Amyloid is not the entire story, but it's a really important part of the story.
Yeah, great question. So the question is whether some of these interventions with blood can also be extended to a disease condition. And those are actually ongoing studies that we have, as well as other labs in the country are actually doing it with things such as Alzheimer's models, Parkinson's models. So right now we don't have the data to give you the answer yet, but those are definitely ongoing.
So the question is, what about supplements? There's a lot of supplements on the market. They say that it'll improve your memory. A couple things, and we actually dealt a lot of this with our IOM report. Just to remind us all that supplements aren't regulated by the FDA, right? Because they're not going after a particular disease. They just say, this will improve your memory. It's not a disease. And the problem is that they don't have to show much evidence, right? You don't have to have any studies. It could just be something somebody, thought sounded good, or, and that often the manufacturing is not regulated either. So you can have varying degrees of whatever ingredient is in there. All to say, again, be caveat emptor, you have to be careful. But, the truth is some supplements have turned out to be quite helpful in many diseases. So I don't want to say they don't work. I think it depends a bit on the supplement, and we need to study them. And a lot of the times people make claims, that the manufacturers make claims, and there's very, very little evidence and varying quality of what you're actually getting. And in fact, there was just a week ago, a big study showing that many people are ending up in the emergency rooms because of the supplements, not necessarily for the brain or for cognition, but because of all the supplements we take. It turns out there are a lot of side effects, so be careful. I didn't have time to get into every risk factor. I would say that diet, again, needs a lot more study. Of all the diets, I do think that the Mediterranean diet is the one that's probably looking like it has the best evidence to maybe be helpful for brain health as well. But in my mind, it's not as well along as some of the things that I showed. So I think preliminarily it does seem like it might be helpful, but we need more research. I think the idea, a lot of it has to do with better vascular, so better cardiovascular and better vascular health in the brain. So they've done some work showing, looking at the MRIs on the Mediterranean diet. There's less of the white matter changes that Dr. Kramer mentioned. That's probably the biggest one. Maybe less inflammation. Yeah, so good question, and everybody wants to know what. I don't think we know, because it's impossible to study every little thing, right? And in all the different people, I think there's some general principles. The general principle is you feel you're learning something new, and it's somewhat challenging. But again, as I mentioned, it has to be something you enjoy. You can't make yourself. I don't like crossword puzzles. I never will like crossword puzzles. And if you said I have to do them, I won't do them. So, but so you have to do something you enjoy, but you do feel that you're learning something new. So I think absolutely listening to music, going to movies, why not?
I mean, I didn't talk about antioxidants at all, but there's a lot of, resveratrol is a great antioxidant, and grape skins and red wine are full of it. Maybe that's a good point on which to end. Thank you.